Before we begin Chapter 14, let's do a few review questions from last time. Which of the following does not have antimicrobial actions? Is it A, silver, B, copper, C, mercury, or D, iron? The answer is D, iron. A lot of microbes actually use iron for different functions. So we can see that silver, copper, and mercury, all heavy metals, have antimicrobial actions. So the answer for this first question is D, iron. Food preservatives include all of the following except A, antibiotics, B, organic acids, C, heavy metals, D, nitrites and nitrates. The answer is C, heavy metals. You do not want to ingest large quantities of heavy metals, so we do not introduce heavy metals into our food for food preservation. But antibiotics are used, organic acids like sorbic acid is used, and nitrites and nitrates are used in preserving lunch meats and bacon and different types of deli meats, things like that. There is currently no antimicrobial growth agent that counteracts all problematic organisms. I will definitely have to fix that typo, but we're talking about the antimicrobial growth agents. And there is not one antimicrobial growth agent that counteracts all problematic organisms. The answer to this question is true. There is currently no antimicrobial growth agent that counteracts all problematic organisms. Some antimicrobial growth agents are good for bacteria of certain types of species. Some are good for fungus. Some are good for different types of microorganisms. But not one single type of antimicrobial growth agent is good against all problematic organisms like endospores or mycobacteria especially. So let's go on to Chapter 14, Antimicrobial Drugs. Chemotherapeutics is an all-encompassing word meaning chemicals that can treat, chemicals that are used for therapy. It's all kinds of different types of medications and treatments that are used to treat diseases and infections. When you see the word chemotherapy, you might be thinking of the treatment for cancer. And yes, that is definitely true. Chemotherapy is a form of treatment for cancer. In this class, we are going to look at the word chemotherapeutic and chemotherapy as an all-encompassing word that includes all kinds of different treatments, antifungals, antibiotics, chemotherapy, yes, and all kinds of different things that are used for treatment. Now, it used to be when someone came down with pneumonia or influenza, it was often fatal because years ago we did not have chemotherapy available to treat these types of infections or diseases that could occur. And those infections and diseases that were once fatal are now treatable because of the development of all kinds of different chemotherapeutics. In the past, physicians could only order bed rest or supportive care or fluids. It was very difficult to treat a patient who had pneumonia or influenza or another type of infection that may be treatable today but was not years ago. Early discoveries include salvarsan that was used for syphilis that was discovered by Paul Ehrlich. Also, one of the most famous early discoveries is the discovery of penicillin. by Alexander Fleming. And you might have read the story about how Alexander Fleming did this by accident. He actually wasn't the best at aseptic techniques and did not have the cleanest of labs. And a Staphylococcus plate became contaminated with a mold. And when it became contaminated with this mold, he noticed that in the area of this mold, no staph grew, no bacteria grew. And so he further experimented with this mold that had grown on his petri dishes and discovered penicillin and found that there were some bacteria that penicillin was able to target. So he discovered penicillin by accident, but it's been one of the most famous and one of the best discoveries of chemotherapeutics. Penicillin was first mass-produced in the 1940s, and it was absolutely critical in saving millions of lives during World War II. It was considered a wonder drug. It saved countless soldiers' lives, and it was used extensively in the war. Unfortunately, today, because of an overuse of antibiotics, bacteria are becoming drug resistant. There used to be a time, especially like when I was younger, that antibiotics were given for everything, including the common cold. If you went to see a doctor and you were sick, they prescribed antibiotics. Today, we need to consider the ramifications of this overprescribing of antibiotics, and we're going to talk about that in this chapter. So again, chemotherapy is the use of chemicals or medications to treat a disease or an infection. It's all encompassing. Antimicrobial medications interfere with the growth of microbes within a host. Antibiotics can be broad spectrum or narrow spectrum. When we talk about broad spectrum antibiotics, they can target a broad range of both gram-positive and gram-negative bacteria. When we do further testing and we can identify what is causing a particular infection, what type of bacteria is causing this infection that's making your patient so sick, we can use a narrow spectrum antibiotic. We can target that bacteria. We can put it down to a narrow, more specific range of microbes. They can be gram-positive or gram-negative, but not both gram-positive and gram-negative bacteria. We make it a smaller, narrow window of treatment. Selective toxicity means that a drug can target the specific harmful microbe with minimal or no damage to the host cells. That's your ultimate goal. You want it to be a narrow spectrum antibiotic that specifically targets the exact bacteria that you want to be targeted. And that way, other cells are not harmed in the process. Antibiotics differ in terms of the organism they are or were isolated from. Where did we develop these antibiotics from? Did they come from nature? Did they come from a mold? Were they created in a lab? Did they come from a plant? Where did they come from? They differ in terms of their chemical structure. All antibiotics have a specific type of chemical structure that can be repeated and duplicated. They differ in terms of their chemotherapeutic index. The chemotherapeutic index is a ratio of the maximum tolerated dose of a medication before you reach toxicity. and its maximum effective dose. So you want to have the best and the highest effective dose without creating a toxic environment for the body. Antibiotics differ in terms of whether they create bacteriostasis, whether they put that bacteria on a pause, which we are going to investigate, or is it a bactericidal, which means that it kills the bacteria. We need to consider if it's a gram-positive or a gram-negative bacteria. If it is a gram-negative bacteria, then most likely we want to create an antibiotic that will cause bacteriostasis. We want to stop that bacteria from replicating. We want to put it on pause, but we do not want to kill it. If we were to kill it, we could release the endotoxin that is in the LPS layer, and that endotoxin could cause extensive difficulties for the patient. It could even send the patient into sepsis, and we will talk more about this later on. Again, we need to consider whether the antibiotic has a broad spectrum or a narrow spectrum. The broad spectrum can affect both gram-positive and gram-negative. It can do a wide range of microorganisms. A narrow spectrum is more targeted. It will be just specific types of bacteria, or just gram-positive bacteria, or just gram-negative bacteria. It's a very narrow spectrum. We need to consider the pharmacokinetics of an antibiotic, how this antibiotic is absorbed, its tissue distribution. We want to know how it is metabolized. How does the body react to this antibiotic? How can the body excrete or eliminate this antibiotic? Sometimes medications can be very hard on the kidneys. And also, as people age, their organ systems don't work as well, and that means their kidneys may not work as well. So we have to consider the pharmacokinetics of an antibiotic. We also need to consider if the patient has comorbidities that can affect the pharmacokinetics of an antibiotic. We need to know the different sites of action of antibiotics. They differ vastly in what their targets are of that bacteria or on that bacteria. We need to look at the interaction with other medications, with other agents, with vitamins and minerals or herbs that you may be taking. Antibiotics differ in how they react with other types of things that you ingest. We also need to consider that antibiotics differ in their potential for the evolution of resistance. Bacteria are becoming resistant, and we have to consider this. We also need to look at the differences in mechanisms of toxicity. Are they selective? Are they not selective? That is really important to understand when we look at antibiotics. And also the cost. Sometimes the cost can be high, and it is difficult for a physician to want to prescribe a high-cost medication for a patient. And then you kind of wonder if the low-cost medications, the low-cost antibiotics are prescribed too frequently. You have to consider the cost as well. This is an excellent image from your textbook showing the action of antimicrobial drugs. Where on that bacteria cell are they targeting? Are they targeting the plasma membrane? Are they targeting within that cell? Are they targeting the metabolic pathways or the DNA synthesis or the RNA synthesis? Are they targeting cell walls or ribosomes? Antibiotics and antimicrobial drugs are developed to have specific targets for parts of specific types of cells. They want to have a target. There are certain things that medications are developed to do. We are going to talk about a few of these. One classification of antimicrobial medications are the cell wall inhibitors. They are the medications that target something about that cell wall. If you remember, penicillin likes to target that peptidoglycan. It likes to target those NAG and NAM cross bridges. And because gram-positive bacteria has so much peptidoglycan that penicillin can attack, it's very effective against gram-positive bacteria. What's interesting about the cell wall inhibitors is they all contain a beta-lactam ring, which you're going to see on the next slide. They all contain that beta-lactam ring, but they also have different types of R groups. They have chemical changes to the R groups, and it can provide different types of activity, but still targeting that cell wall. It can change the acid stability. It also helps against the resistance to beta-lactamase degradation by changing those R groups. If we can adjust those antibiotics, changing the R groups on the beta-lactam ring, maybe we can improve these cell wall inhibitors so that the bacteria don't recognize them as much. So if you look at this slide, you can see that in the center of all of these different types of antibiotics, you can see that they all have this particular beta-lactam ring. But all of the different R groups are attached to that beta-lactam ring, making it a little bit different. Still a cell wall inhibitor, still attacking something about that cell wall, but doing it in a different way, and maybe doing it with more selectivity, maybe potentially doing a more narrow target. MRSA is an infection that we will talk about a few times during the course of this class. It's methicillin-resistant Staphylococcus aureus. Now remember, we have Staph aureus as part of our normal microbiome. It's on the surface of our skin. It is beneficial because it can block more pathogenic microorganisms. But many of us are carriers of MRSA. We may not even know it because we do not have symptoms. Cases of MRSA have dramatically increased since the 1970s. It used to be that MRSA was a nosocomial infection that occurred to a patient who was immunocompromised or vulnerable and was in a hospital. But now MRSA is seen in many different types of infections. You do need to have a portal of entry for MRSA, the infection, to take hold. You can see in these images, in the lower image, that was a knee replacement surgery. The sterile field might have been compromised, or somehow, because there was a portal of entry into that knee, an infection developed. And unfortunately, it became a MRSA infection that became out of control. And the patient ended up having to have their leg amputated because of the knee replacement surgery. It can occur from a scratch or a bite or a cut. If it gets into the dermal layer, it can spread throughout the body system. MRSA is a very, very serious infection. Now, if you get a cut or a scrape or something like that at home, go to your first aid kit, and hopefully you have something like topical bacitracin or Neosporin in your first aid kit. that can actually inhibit the cell wall synthesis. It targets the peptidoglycan in that Staph aureus. It can target the cell wall. Bacitracin is a cell wall inhibitor, and it can be very beneficial if you treat your cuts and your scrapes. Now, vancomycin, especially IV vancomycin in the hospital, used to be the typical treatment for MRSA because it is so serious when it becomes systemic. Unfortunately, now they are becoming used to vancomycin, and VRSA is occurring, vancomycin-resistant Staphylococcus aureus. There are bacteria that are resistant to vancomycin, and vancomycin is not helping in the treatment of this very serious infection. So other antibiotics have to be developed in order to combat this very serious infection that can occur. Protein synthesis inhibitors are another classification of antimicrobial medications. These medications work on the inside of the cell. They target transcription and translation. Metabolism within that cell is affected. Replication is affected within that cell. Unfortunately, when you go within the cell and you target things that are occurring within the cell, there may be an increase in side effects. And medications like tetracycline, while they can block the RNA and inhibit protein synthesis, often may have additional side effects that are occurring from the medication, even though they may be beneficial in treating a certain type of infection. So it's great that we have developed antimicrobials that can target specific things about the cell or in the cell or on the cell. But we have to be mindful of the side effects that can occur from the medication specifically, because we are working with how that cell functions. Researchers are always looking for great targets on the cells of microbes that they can interfere with or inhibit or prevent the cell from doing something. And because they're trying to reduce the side effects by targeting certain functions, if they can get very specific within that cell, sometimes they can reduce the side effects and the collateral damage because it's more specific. There are medications that specifically can target just the folic acid, just the metabolic pathway for folic acid. And the synthesis of that, and they can prevent bacterial growth. And because these types of medications can target just the folic acid, they're not causing any additional problems for that cell specifically. And because they're stopping that pathway and inhibiting the bacterial growth, they are able to take care of certain types of infections that way without causing additional side effects. The Antimicrobial Susceptibility Report, or Antibiogram, is a periodic summary of antimicrobial susceptibilities of local bacterial isolates that are submitted to the hospital's clinical microbiology laboratory. It is submitted to these hospital laboratories within healthcare systems, and the hospitals or the healthcare system can develop a chart like this antibiogram, so you can see which bacteria are susceptible to which antimicrobial. It's really interesting, and I absolutely love this assignment that we do. What's also interesting about the antibiogram is that when they look at these bacterial isolates, they're able to monitor for resistance. And on this antibiogram, you will actually see bacteria that are resistant to certain antimicrobials. The antibiogram helps to track the resistance of these bacteria. So when you look at this chart, you are going to look for the largest number. Say the number within that column is 99. That means that that bacteria has a 99% susceptibility to that antimicrobial, right? Think of it that way. 66 is far less than 98. And so you would choose the 98 susceptibility over the 66. So you're always looking for the higher number. That means that bacteria, that percentage of bacteria will be susceptible to that antimicrobial. So physicians can use the antibiogram for direction. If you culture a certain type of infection and you find out what exactly is causing that, what type of bacteria, what is causing that infection in that individual, you could look at this antimicrobial susceptibility report. Look in that column, whether it was for an inpatient or an outpatient. If you look, the data is different. And you need to look to make sure you're at the right bacteria, the gram-positive and gram-negative. So you have to be really careful when you're looking at this report that you look at the right side. There's two sides to this report. So a physician can look for the best possible antibiotic in that column that is affecting that type of bacteria. But the physician also has to consider the patient as well. While they could see that a certain type of antibiotic is really effective, they need to consider if that patient might have allergies or might have comorbidities that might not be able to metabolize that medication well. Remember, we talked about the pharmacokinetics, and if a patient isn't able to metabolize a certain antibiotic, that would not be the right choice for them. You also have to look at the history of infections, or how many times maybe they have had this specific infection, or what antibiotics they've had in the past. So an antimicrobial susceptibility report can be an excellent guide for a physician, but they also have to consider other things when they're choosing that antibiotic. But I really think this is an excellent assignment, and you will enjoy completing it. And you will learn something too. Okay, let's look at antifungal drugs now. We've looked at antibiotics a lot, so let's kind of shift gears a little bit and look at the antifungal drugs. Remember, we have talked about in the past that treating fungus or treating helminths or treating a eukaryote type of infection, one that has been caused by a eukaryote, is difficult to treat for us because they can target components of that fungus or of that parasitic worm that have a structure that's similar to ours. In antifungal drugs, they can target the essential cell wall components. Now, if you look at that word, ergosterol, and you look below, we have cholesterol in our cell walls. If you look at ergosterol to the right, it is very similar to cholesterol. So you can understand how a fungal drug might miss the target and not target that ergosterol, but actually target our cells that has cholesterol. So you can't use high doses that may be required for treatment. Because if you use these high doses and it damages our cells or attacks components within our cells, then it can increase the side effects and it can cause more complications in addition to the infection. So treating fungus and treating helminth parasitic infections and eukaryotic infections is difficult to do. Remember that these drugs have to be monitored very closely so that we can make sure that it is targeting the infection and targeting the microbe that is causing the infection instead of damaging our own cells. Let's talk about the anti-protozoan drugs now. So chloroquine might look familiar to you. Hydroxychloroquine has been in the news, and it is one of the chloroquine medications that are out there. Chloroquine inhibits DNA synthesis. It was developed for treatment of malaria, and it's been used in the treatment of lupus and the treatment of some forms of arthritis. Hydroxychloroquine has been considered as a treatment for COVID-19. Unfortunately, a lot of the hydroxychloroquine that was being used for lupus patients then became scarce, and lupus patients have been having to go without it because they were using hydroxychloroquine to treat COVID-19. And unfortunately, when you don't continue on your medication for hydroxychloroquine, you can have really serious withdrawal symptoms. Because this medication inhibits the DNA synthesis. Remember, when a medication is targeting a function of that cell, it can lead to increased side effects. If you've taken a medication for a while and your body is used to that, your body is being helped by this medication, and then all of a sudden you're taken off of this medication or the amount that you're taking is reduced, it can have serious consequences. There are other anti-protozoan drugs that target different things, like the chelation of iron ions within a cell. See, antimicrobials can be super specific in what they do, and they use quinolines for different types of amoebas and those types of protozoas that cause infections like that. There's also metronidazole, which damages, again, the DNA of a cell. And it is used in Giardia. And that image is actually a picture of Giardia. And we've talked a little bit about Giardia before. It's something that can occur from contaminated water. And you can get an infection from the little protozoa that is in the water. What I want you to see from this slide is how many different types of medications have been developed to treat parasitic worms, and how many different types of targets for those worms those medications can have. They can prevent ATP generation in tapeworms so it doesn't have the energy. It can alter the membrane permeability in flatworms. It can block the neuromuscular junctions in intestinal roundworms and inhibit nutrient absorption in different roundworms. It can paralyze worms. There's all kinds of different things that anti-helminth drugs do. What you should also consider is that there is not one anti-helminth drug for all types of parasitic worms. You cannot take a medication for a tapeworm that also will affect a flatworm or a roundworm. They are very specific to the type of parasitic worm that these medications treat. Antibiotics cannot be used to treat viruses. Viruses are very different from bacteria. You know that a virus has to have a host in order to replicate, and their outside structures are very vastly different from one another. Acyclovir is a medication that was developed to treat some viruses, and we will talk about those specific viruses that acyclovir treats in the last part of the class. But what I want you to know is that acyclovir is not good for all viruses, right? Because the viruses are so different. But what it can do for some viruses, it can bind to the viral DNA polymerase and cause a disruption of the DNA replication. It stops the growth of that virus. Somehow, it will attack that life cycle of that particular virus that it's effective against. Remember, that virus has all of those steps that it has to complete. It has to complete the attachment, the penetration, the biosynthesis, the maturation, the release. This drug can target a part of that life cycle and is able to stop the replication of that virus. Drug resistance is a very important topic that we need to discuss. Bacteria are becoming drug resistant. They are becoming drug resistant for a number of different reasons that we are going to talk about in the next few slides. And I want you to be able to understand how important it is that we must consider the drug resistance that is occurring in our world. And you will be doing a reflection paper on drug resistance after watching a video, and you need to consider these different things that are happening. So, a variety of mutations can lead to antibiotic resistance. Now, these are specific to the bacteria themselves, the mechanisms of antibiotic resistance by the microbe. So these can include the enzymatic destruction of the medication. So when that antibiotic comes towards that bacteria, they are able to produce enzymes that destroy the drug before it can penetrate. And they can prevent the drug penetration. They can release different types of enzymes or chemicals or alter their drug's target site so the drug cannot penetrate at all. It can't bind to the bacteria, so there's no way it can penetrate. So it prevents drug penetration, it can alter the drug's target site, and it can just rapidly reject the drug. If the drug does make it to attaching to the bacteria, it finds a way to just reject it and expel it from its surface. Resistance genes are often transferable DNA. Remember, bacteria have that pilus. Some of them can have that pilus, and they can attach to another bacteria, and they can share their drug-resistant genes with that bacteria. And remember, it's also not species-specific. A bacteria can share these genes with an archaea. So we can share this drug resistance with different types of microbes through this pilus, through horizontal gene transfer. These bacteria can exchange these genes and pass them on to bacteria or microorganisms that weren't previously resistant to this particular type of antibiotic. So, drug resistance is a very serious concern. So these were things that were specific to the bacteria, specific to the microbe. Let's talk about the human reason for bacteria becoming drug resistant to antibiotics. The misuse of antibiotics is the leading cause of drug resistance. So, using outdated or weakened antibiotics. If we are prescribed an antibiotic for a particular infection, and we just take that antibiotic until, I'm think I'm feeling better. I'm going to stop taking it and I'm going to save the rest. So you save them up in your cupboard, and then later on, maybe a year later or 2 years later, you get another type of infection and you remember you have those antibiotics in your cupboard. So you take those for this new infection that you have, thinking that all antibiotics will treat all infections, but in actuality, they don't. So using outdated and weakened antibiotics that you have hoarded over the years is one of the misuses of antibiotics. Using antibiotics for a common cold or other condition that does not warrant antibiotic treatment. That goes along with using antibiotics to treat viral infections. The common cold is a virus. You cannot treat the common cold with an antibiotic. You cannot treat a virus with an antibiotic. But patients used to go see their doctor, and the doctor would prescribe an antibiotic for whatever infection they were having, whether it was a virus or not, because patients go in and they want a prescription. They want a cure. They want to be helped, and physicians were compelled to do this. They wanted to have that satisfaction from the patient, and so they would write these prescriptions. They also didn't consider drug resistance. It wasn't a consideration in the future. They knew they had these antibiotics and they knew that they could help some things. So they would give them to patients, not knowing that they were creating future drug resistance. The use of antibiotics in animal husbandry. We've talked about this. Animals are injected with antibiotics to help them grow bigger so that they can produce more meat. They inject antibiotics because they want to limit the exposure to that meat that might have been exposed to the bacteria from the cows or the chickens living in bad conditions, and so it's used in animal husbandry. But if you eat foods that have previously been injected with antibiotics and you get a low dose of that antibiotic into your system, and if you continue to add this low dose of antibiotic to your system over the years, then this can also lead to some drug resistance. We talked about the cost of antibiotics, low costs or high cost antibiotics. When the costs were very low, and we go back to that scenario where the physicians are just writing out prescriptions of low cost antibiotics, it was a cheap way to give a pill to a patient. Patient satisfaction scores. It was really important for these physicians to have these high patient satisfaction scores. Yes, this doctor gave me this medication. This medication, maybe it helped. Maybe they were actually getting better when they went to the doctor before they had the opportunity to receive a prescription, and the prescription really didn't have an effect on them getting better. They were already getting better, but they attribute it to the doctor giving them this pill that cured them. So patient satisfaction scores are something that physicians used to really focus on. Also, if you go back to remember, we talked about the broad spectrum antibiotics and the narrow spectrum antibiotics. If you take a broad spectrum antibiotic, it's going to kill gram-negative bacteria, it's going to kill gram-positive bacteria, it's going to kill the bacteria in your normal microbiome. Your normal microbiome has these excellent bacteria that are trying to help protect you as well, keeping out the pathogens. But if you take a broad spectrum antibiotic that wipes out all kinds of different types of bacteria, from gram-positive to gram-negative to your normal microbiome, you're destroying the good bacteria too. Not just bad bacteria, but good bacteria. And unfortunately, then that leaves an opening for the pathogens to come in addition to allowing the drug-resistant pathogens to proliferate and causing super infections because they have more places to be within the body. So we have to consider the fact that when we take these broad-spectrum antibiotics, that we can be doing serious harm to our normal microbiome and killing off more bacteria than we need to. Narrow-spectrum antibiotics are the way to go. Often when you go in with a very serious infection, you may start with a broad spectrum, but lab cultures will be done, and then you can be removed from that broad spectrum antibiotic and put on a narrow spectrum antibiotic to reduce the death of all that good normal microbiome. Here are two definitions about the effects of drug combinations. Synergism occurs when the effect of two drugs together is greater than the effect of either alone. So oftentimes we will combine medications to have a better effectiveness on whatever the infection is we are trying to treat. Those medications are okay by themselves, but when they are combined with another type of medication in a combination that has been determined to be effective, the synergism that occurs can make them really work together and be much more effective than if they were given to you separately. But there is also antagonism that can occur. Antagonism occurs when the effect of two drugs together is less than the effect of either alone. So you could have a great medication that you're taking, and then you add in another medication that might make the medication you were previously taking less effective. When combined, they interact with each other, so the effectiveness is decreased. That's antagonism. They do not work together. They are less effective. They are much better when they're separately and not taken together. Classic vaccines imitate a natural infection within the body, and they are developed using weakened or dead microbes or toxins or particles. And these microbes or toxins or particles can activate the immune system to retain the memory of that particular infection. So we can activate the immune system. They remembers this infection that they had. These microbes were introduced. They were these foreign things that were introduced, and the immune system remembers what they are. So then when that infection comes along, when you encounter it in your daily life and it enters into your system, your immune system, because it's had this vaccine, can recognize it and target it and work to get rid of it. These are the three classic vaccination methods that I want you to know. We have weakened microbes that are used in vaccinations for malaria and measles, mumps, and rubella and tuberculosis. These weakened microbes can sometimes cause mild symptoms when you receive that vaccination because they are weakened. They're not dead. So it is possible that you will have mild symptoms, but you will not have the full-blown infection of malaria or measles, mumps, or rubella or tuberculosis. But you could have some mild symptoms because your immune system is identifying it as something that is foreign, that is possibly going to make you sick. You can also have vaccines from using dead microbial shells. These are dead shells of the microbes. They trick the immune system. The immune system can see them and know that they are something that's foreign that they need to attack or need to remember to attack. Also, they need to develop that memory. And those can be from influenza vaccines, cholera vaccines, rabies vaccines. They can trigger a short weakened immune response when you encounter that infection. So you have to receive these multiple times. You have to have a variety of these vaccinations, maybe yearly in the case of influenza. You can also have weakened toxins. There are no microbes in the toxins. These are specifically taking the toxins from tetanus and diphtheria and using those weakened toxins to develop a vaccine that your immune system can identify and recognize. And again, you may have a few mild symptoms. This also triggers a short, weak immune response. So you have to have multiple administrations of this vaccine. You have to have boosters, like in tetanus. New vaccine research is being done for a number of different types of infections. You can have an adjuvant or a conjugate vaccine, which combines a weak antigen with a strong antigen to cause an even stronger immune response. HPV, which we will talk about later in the class, human papillomavirus. You might have heard of Gardasil. Gardasil is a conjugate vaccine. Why it's important that we have this vaccine to target human papillomavirus is because there are a few strains that can cause cancer. This research is also being done to develop a vaccine for hepatitis. There are also DNA vaccines, and those are in the news right now, along with RNA vaccines, which I'm going to talk about both of these in relation to COVID-19. So for a DNA vaccine, one way you can use a DNA vaccine is to inject genetically engineered DNA for an inherited gene, an oncogene, or a microbe, hoping that you can somehow change how that cell is responding. It's being used for HIV, breast cancer, or COVID-19. These newly injected genes can cause interference somehow with cell function. In the case of COVID-19, Providence Cancer Center is researching a vaccine called Corvax 12. And this is an encodable DNA vaccine which you can inject, and it's designed to cause the immune system to destroy the spikes. You know, you've all seen that picture of COVID-19 that has those spikes all around the outside. And what this vaccine is to wants to help the immune system to destroy those spikes, to wipe out those spikes so COVID-19 can't use those for attachment. Currently, there is a clinical trial going on with Kaiser Permanente of Washington in Seattle in conjunction with the National Institute of Health. And they are trying to develop a vaccine that is specific to messenger RNA. This vaccine will direct the body cells to express a viral protein that will elicit a very strong immune response against the COVID-19 virus. It has shown excellent promise in animal models, and we hope that these first clinical trials will be successful in humans. Because bacteria are becoming drug resistant, we have to start looking outside of the box. We have to start researching other things, maybe in nature, in plants, and in animals that can be beneficial for us, that can help treat drug resistant bacterial infections. If you look back at the bacteriophages, remember bacteriophages are viruses that attack bacteria. You can identify a specific virus that can attack a specific type of bacteria. You would be able to figure that out in the lab. But the difficulty comes in when you need to convince a patient that you want to introduce a virus into their system in order to remove a certain type of bacteria. Especially in this age of COVID-19, where we have seen so much and been affected by so much from this virus. It might be a very difficult conversation to talk about the therapy that can be done using bacteriophages. You can see from this image that is in your textbook how the approval of new antimicrobials has dramatically decreased over the years. It takes quite a bit to develop an antimicrobial. It takes a lot of money. It takes a lot of things that need to be completed before a drug is approved by the FDA. With COVID-19 and all that is occurring, I hope that this trend does not continue in this way. I hope that they can see the value of new medications being developed, new treatments being found and put on the market that can help with drug-resistant bacteria and viruses, novel viruses that are occurring. We need to find a way to increase the research into the development of new antimicrobials. And I do hope that things will change in the future. Before COVID-19, MIT was looking at developing a new antiviral that is called Draco. You don't need to know that long, fancy word. But it is an antiviral that can cause infected cells that have been infected by a virus to commit cell suicide. It can cause apoptosis. Infected cells produce this viral RNA, right, while they are multiplying within that cell. And Draco can bind to it and initiate apoptosis. It has shown promising effectiveness against rhinoviruses, which are one of the causes of the common cold. It's amazing that in 2020 we have not been able to find a virus that is effective against the common cold. But this has shown some promising effectiveness for rhinoviruses. It has also been shown to be effective in some cases against H1N1 influenza and against dengue fever. So research is being done, and it has continued before COVID-19, but not as much as we have hoped. With antibiotic resistance on the rise, we need to start thinking outside the box, and we need to start looking to nature. There have been many different types of research studies thinking this way and trying to use natural things in order to treat infections. They have been looking at alligator blood. Alligators don't get many infections, and if you think about it, they live in these dirty, swampy, nasty areas, and they are doing just fine. So they've been looking at alligator blood to see if there's something within that blood that is able to control microbial growth and be used as an antimicrobial. And frog skin. If you think about it, frogs have this moist, soft skin that is the perfect place for bacteria to grow. And so they've been seeing how frog skin might be a possibility. They've been looking at Lipitor, which is a cholesterol drug. Some microorganisms use cholesterol for energy, and if they're able to get rid of that energy source, could they potentially inhibit the growth of that microorganism and use it as an antimicrobial treatment? We need to also look to animals. When animals get sick, what do? If a horse has a stomach ache or a cow, Or a dolphin. If they get sick, what do they do to treat themselves and to help themselves get well? So we need to think outside the box, and we need to start looking at nature and different types of remedies that we can use for treatments and therapies against these antibiotic resistant infections that have been developing. Researchers found that shark skin can repel some microbes. I mean, think about that sharp edges of the shark skin. It was discovered when scientists were trying to prevent algae from attaching to the surface of submarines and boats, and they put down a layer of shark skin, and no algae grew on those surfaces. And again, more things in nature can have certain types of surfaces where no microorganisms will grow. And they have been doing research on gecko skin and cicada wings as well. There are more things that have been considered as antimicrobial. Essential oils have been used for centuries for their antimicrobial properties. In the 15th century, folklore tells a story about how these thieves who would rob the bodies of those who had died from the plague would not get sick. And it was because they had developed this combination of oils, including lemon and clove and thyme and all of these different types of essential oils together. And they had covered themselves in this oil, and they did not get the plague. And that's where thieves' oil comes from. You can interfere with quorum sensing. Quorum sensing is how microorganisms communicate with each other, and you can disrupt that communication so that they are not able to form biofilms and things like that. You can look at using very low trace amounts of silver in antibiotics to increase the effectiveness or cause synergism to occur between the silver and the effectiveness of that antibiotic to increase it, right? We want to increase the effectiveness of the antibiotic by adding small doses of silver. But remember, not a lot of silver because it can be toxic in large amounts. And we can develop biofilm penetrating peptides that can break apart the biofilm and disrupt that community of bacteria. One of my favorite things that's being researched are called snaps. And they are like little ninja stars that you can throw at bacteria. So they've developed these nano-engineered peptide polymers that are like these little stars, and they can shred apart the walls of the bacteria. They can tear them open. And they've tested these snaps on E. coli and Pseudomonas and Klebsiella pneumoniae. And I think it's really interesting research. So we need to think about these drug-resistant bacteria and what we can do if we do not have effective antibiotics to use in the treatment of these. Antibiotics are losing effectiveness. A number of them have lost their effectiveness. Think about MRSA. MRSA is becoming VRSA because it is now resistant to vancomycin. It's just continuing. So we need to think about what is treated now can become life-threatening in the future because of the loss of effectiveness of certain antibiotics. Some gram-negative antibiotics are becoming ineffective against some gram-negative infections. We are not able to control gram-negative infections that can release those toxins and cause very serious infections. And there's continuing speculation out in the world about how we can practice medicine without antibiotics. We have to start considering different things in our world. And how do we encourage research and development? What do we do to encourage research? A serious pandemic like COVID-19 should not be something that brings to light that we are seriously lacking in how we treat antimicrobials. We have got to find better ways to encourage the development of antimicrobial medications. Also, we need to research more things, herbal remedies and things outside of the box that we might not have considered before. Here is a question to ponder. Why are microbes becoming resistant? And what do you think modern medicine should do about it? We know that bacteria are evolving. We know that there are viruses out there that could be even far worse than COVID-19. We have to consider these things. We need to be prepared. And I think that COVID-19 might have been a wake-up call for some of these things that we need to consider. We need to develop vaccines, and we need to develop better treatments for the microbes that do cause disease. But remember, not all microbes cause disease. But yes, there are some serious ones out there, and we do need to think about that. And we do need to think about it for the safety and health of our world populations in the future. And that's the end of chapter 14.